Nah, mungkin tanpa berlama-lama lagi ya, jadi saya akan membacakan CV dari Pak Matias Steinberg. Jadi beliau memiliki beberapa background yang cukup luar biasa ya. Jadi tahun 92 beliau mendapatkan diploma degree-nya dengan tesis di Natural Science atau biologi dari Westphalian Wilhelms University, Münster, Germany. Lalu beliau juga melanjutkan sebagai researcher di Institute of Applied Ecology at Westphalian Wilhelms University, Münster, Germany. Lalu setelah itu 93 sampai 96 beliau juga menjabat sebagai researcher di European Commission Environmental Institute Water Resources Management Unit di Ispra, Italia. Lalu dari tahun 97 sampai 2013, nah beliau tuh sudah mulai bergabung di Partec dengan beberapa fungsi yang berganti ya. Jadi sempat menjadi International Sales dan Marketing Manager Research dan Industrial Flow Cytometer, lalu sempat juga menjadi Product Manager untuk bidang Research and Industrial Flow Cytometry, lalu Application dan Technical Specialist Flow Cytometry, dan juga sempat menjadi Field Application Engineer untuk Flow Cytometry juga. Nah, setelah itu beliau juga sempat menjabat menjadi Product Manager untuk Research dan Industrial Flow Cytometry di Sysmex Partec. Lalu berikutnya beliau juga pernah menjadi Manajer Akademi dan Product Manager IFCM di Sysmex Partec. Nah, untuk current role-nya sendiri yang sekarang beliau menjabat sebagai Manajer Product Promotion, Akademi, dan Support di Sysmex Partec. Nah, mungkin tanpa berlama-lama lagi, Matthias, are you ready? Hello, Matthias.
Yes, hello.
Yeah.
Let's check. Yeah, okay. Yeah, hello from my side. Thank you for joining today the meeting in the webinar about the fitness of pollen. Stephanie, shall I go on and proceed with the talk right now? Hello? Okay, yeah, so welcome, and let me share my screen so that we can directly enter the topic of today's talk, which is the pollen viability. Just a second, I have to redo it. Okay, please give me a sign when you can see the presentation. Is that okay? You can see it? Hello? I don't get any
hello,
feedback.
Matthias. Sorry for the technical problem. Yes, we can see your screen.
Okay, good, great. So then let me start with the talk about the analysis of pollen fitness by flow cytometry. Yeah, now, what are we going to talk about today? So I will have 3 chapters in my presentation. So first, I just will explain very briefly what is the pollen viability. Then I will describe some common traditional methods to determine pollen viability. And in the end, I will explain how Sysmex is doing pollen viability and what is our proposal and technology to determine this property of pollen. Now, what is the pollen viability? So viable pollen is pollen that is competent to deliver 2 male gametes to the embryo sac. So the fertilization of the egg is, of course, the final goal of the pollen. But in generally, the term pollen viability is used as a common term describing the capacity of pollen to live, to grow, to germinate, and to develop, because the pollen grain during its life cycle has different phases and needs to perform different actions. So the first one is the pollen grain has to attach on the stigma on top of the style of the female organ of the plant. Then it has to hydrate in case it is dehydrated, which often is the case, and to germinate. So this can also be defined as a germinability of the pollen grain. And then it has to produce the pollen tube to grow through the entire style in order to reach the ovary, release the sperm nuclei, and then finally fertilize the egg cells and the endosperm and the polar nuclei for the production of the endosperm. The performance of the pollen grain is related to different actions: penetration of the stigma, growth through the style, enter the embryo sac, and of course release the gametes. Now, why shall we assess pollen viability? So the pollen itself, of course, plays a major role and is important for the species development, the dispersal, the fitness, and the survival of the next generation. So it's mediating sexual reproduction. And for plant breeders, of course, pollen must be viable at the time of pollination in order to ensure for seed or fruit production. Some examples can be, for example, pollen in storage for germ plasma conservation, or if hybrid breeding is done with different plants flowering at different locations or time points, then you need to store pollen, you need to transport them. Of course, during these processes, the viability of pollen can be affected and influenced. So the pollen viability in general is influenced by the environment and also by the genotype of the pollen itself. The quality of pollen, and quality means here viability, varies by time, by the nutrition status, also by environmental climate condition at maturation and storage. And of course, for several pollen, it is known that post-maturation preconditioning plays an important role for them to be functional. Now, what are classical methods to determine pollen viability? So the easiest and most straightforward method is just looking at them. And this is a direct observation by light microscopy. So here the morphological status can be observed. And just by looking at the pollen grains, someone can differentiate abnormal pollen grains, which are then suspicious in order to define them as potentially non-viable. So this is a quick and of course mobile method. You just need a very simple microscope for that, and gives you a first overview of the pollen quality. But of course it's absolutely non-specific, and it's just judging very subjective the appearance of the pollen grain. Then there are a series of different other microscopic techniques which are based on staining of pollen and staining of different parts of the pollen. So, for example, the cytoplasma and the nuclei can be stained to indicate if there are intact membranes or intact nuclei in the pollen grain. Other staining tests with iodine, potassium iodide, or aniline blue are staining other parts of the pollen, other chemical components, and usually the absence of color stains indicate limited viability or non-viable pollen. Of course, all these microscopic tests are limited to a relative low number of pollen which can be observed in a reasonable time, and the counting of higher numbers involves quite a big workload, and of course interpretation is always very subjective depending on the operator and the conditions. Another often used test is the so-called microscopic fluorochromatic reaction test, FCR, which has been developed by a couple, Heslop and Harrison, in the 1970s. So it was published first in the 1970s. And this is a first test which is really reflecting metabolic activity inside the pollen. It is based on a substance called fluorescein diacetate, or FDA, which is non-fluorescent and is capable to penetrate living membranes. And if it enters the pollen grain, the cytoplasma of the pollen grain, it is cleaved by esterases, non-specific esterases enzymes into a fluorescent component, the fluorescein, which loses the capacity to penetrate membranes, and therefore this fluorescent dye, fluorescein, is accumulated inside the pollen, the cytoplasma of the pollen, and it makes, after fluorescence excitation, the pollen shine green. This test is relatively simple and fast, and many papers after this initial publication have confirmed a good correlation of the results with other tests of pollen fitness and viability. Anyhow, still it's a microscopic analysis. The counted numbers of pollens are very small, or are relatively small usually, and it requires a subjective interpretation of the fluorescence intensity. Here on the left side, you see 3 examples made from the same sample. You see pollen grains shining very bright green, medium, and very dim green. Of course, here the transitions between different states are fluent, and here the test depends a lot of the status of the illumination, how the pollen grain is positioned to the focal level of the microscope and the focus of the excitation. So it's quite a subjective work, and of course analyzing fluorescent stain samples in a microscope and count particles is quite an intense and sometimes even annoying work. One limitation is connected to this type of test. If we have pollen which is in the state of dehydration, when it usually has a porous plasma membrane, then of course the fluorochrome cannot accumulate inside the pollen grain, and it will leave or leak out of the pollen grain, and a strong fluorescence signal cannot be expected. Okay, next possibility to test pollen viability. So there are several tests doing or observing the behavior of pollen grains on the female part. So the common test is a so-called seed set or artificial pollination, and this measures the capability of pollen to fertilize a plant. So here in the seed set, female stigmata, so vital female stigmata on the plant itself are pollinated with test pollen, and successful pollination is monitored by looking at the fertilized ovaries after some days. Of course, here we do not look on the individual pollen grain because there are always several pollen grains put on the stigma. Somehow it is possible to also get an estimation of fertilization after this treatment. There are many different factors contributing to the success, amount of pollen which is put on the stigmata, the capacity of the stigmata to be receptive even by the treatment itself, it can become unreceptive for the pollen. Then we have compatibility questions between pollen and stigmata, and different other factors can affect the success of this. This is a possibility really to investigate on more factors, especially in the combination pollen grain and female part, if this is working, but it does not give us really a good clue on the fitness of the individual pollen. Next to this seed set, there are similar in vitro germination tests where we expose pollen grain to media in an experimental setup, and we observe if the pollen is developing inside the media. So there are a bunch of different media described. The most frequently used is from Brubaker and Quark from 1963, so quite old. But there are plenty of modifications even for specific species, because not all pollen grains are growing on one particular media. So there are even some species which cannot grow on media at all. For wheat, for example, this has been described. Okay, and as a, let's say, last example of traditional tests is the in vivo germination, or so-called semi-in vivo germination, also known as stigmatic germination. Here we do similar as in the pollination. We excise female parts and put them in a specific media, or we do that on the living plant and then extract the female part after pollination and observe with fluorescence staining if the pollen tube has been developed and grown into the style. So that, of course, is quite tricky handling on individual stigmata. It's quite work intense, and of course here we are facing again the question of incompatibility. Okay, yeah, as a conclusion on these tests, well, all methods of testing depend on variables, and no one is really able to confirm that a particular pollen sample is inviable and will not fertilize. So we get indications, but it's not really a proof. Yeah, so that means tests are estimations and produce likelihoods. But for most questions, I think this is sufficient result. Usually they are not suitable as a field test, and yeah, different factors influence the information which we can gain. And all of these tests underlie a really subjective interpretation of the data. Anyhow, the testing of pollen has a value for research and breeding in agriculture, and delivers still information about plant fertility and some, and is useful for certain questions to support here decision making. Okay, now what is our way? Let me briefly describe what we offer as a Sysmex Partec. So we have a kit, a reagent kit, which is called Pollen Viability Kit. And this is a kit which contains staining solutions for fluorescence staining, two different of pollen. And it's also applicable to microspores, to extracted microspores for subsequent analysis in a flow cytometer. So the kit contains these two solutions, solution A and B. And what is behind A and B? One solution A contains the substance fluorescein diacetate, which we know already from the Heslop-Harrison test, the fluorochromatic test I described before. So this is this non-fluorescent substance which penetrates into the cytoplasma and is cleaved by esterases and accumulated in the cells. So that is this situation here. So the non-fluorescent fluorescein diacetate molecules go into the pollen grain, are cleaved, and then the pollen grain becomes shiny green. And the second dye is called propidium iodide, and this is a non-membrane permeable dye which is staining the DNA. So that's this red dye here. It only goes into nuclei, into pollen grains, and stains the nuclei, which are which have a damaged membrane. So practically dead pollen. Next to the DNA, it is labeling also staining the pollen grain wall on a certain magnitude, so that the dead pollen are shining red but not green. Okay, so here we test two properties of the cellular viability, the membrane integrity and the metabolism. So how is this looking like and how is this done? The procedure is just very simple. We take a tube, a test tube, a plastic test tube, and suspend a certain number of pollen in 2 milliliters of PBS or a similar buffer, which is maintaining the viability of the pollen. We add the solution A, mix and incubate for a short period, 10 minutes at room temperature. We add solution B, the propidium iodide, mix again the sample well, and then the sample is ready to be analyzed by the analyzer. And here on the left, you see 4 pictures from fluorescence or light microscopy. So first we have here an unstained date pollen, date palm pollen. Then we have here a single viable pollen. You see the shining green cytoplasmatic fluorescence and the light red fluorescence of the pollen grain wall. We have dead pollen too. One here, you see it has a shining fluorescence all over, and we see 2 nuclei here which are red shining, and of course the pollen wall. So this would be a pollen with a defective membrane, still having intact nuclei, shining bright red, but no green fluorescence present here. And the other one here has even no nuclei anymore, so it's effectively only an empty shell of a pollen grain. It just gives us the fluorescence of the wall, the red fluorescence of the wall. And here we have a mixture of different, straightforward green fluorescent living pollen grains, like this and this. We have here a case where we still have nuclei, the 2 nuclei, so we have a red fluorescence and no green fluorescence. This one here is completely empty. It has just a red pollen grain wall. Okay, yeah. What do we need else? We need an analyzer for that, and here Sysmex Partec offers different type of analyzers, different type of flow cytometers, so which are fulfilling the requirements, the technical requirements. So the names are Cyflow-Cube 6, Cyflow-Cube 8, which provide a laser light source of 488 laser light source, which is capable to excite the 2 fluorochromes, and it has analyzing parameters for scattered light and red and green fluorescence. That is what we need. Next to these cube instrumentation, we offer also other instrumentation like the Cyflow Space, which is also capable to carry out and support this test. Now, how the result is looking like. So here you see a typical result, and this is made by sunflower pollen. So just by displaying the green collected fluorescence of the fluorescein versus the red fluorescence, green fluorescence versus red fluorescence, we can depict each individual pollen, which has been analyzed as a blue dot. Yes, so here you see single blue dots, and then when we go to the center of the population, we get accumulation of different dots to bigger dots. When we have overlaying dots, then the color is turning into green or yellow and red. So a red area is indicating a high number of individual measured pollen grains on top of each other. So basically we can discriminate two major populations, which are mainly red fluorescence, which are the non-viable pollen grains, and which are green and red fluorescence, which are the viable pollen grains. There are some in between, so these are grains which still have some metabolic activity, but have also already defective membranes, so they are on the way to from viable to non-viable. And you see, we can deal here with high numbers, 10,000 pollens. So that is really gives us a statistical and really objective interpretation of the data. To show you the procedure, the ease of the procedure, I just will now show a small video of about 3 minutes, which is displaying the procedure from the beginning to the end. I hope you can hear the sound. Audible.
Yeah, Matthias, we can hear the sound.
Of course, that's here a very simple way to harvest pollen sacs. Sunflower is a very nice sample because everything is really big and can be made visible very easy. So that's the extraction buffer. The pollen grains are suspended, released from the pollen, from the anther. Okay, to prevent that the analyzer later on is blocked with big parts from the plant. We filter so that we get we filter the sample so that we get a clear suspension without other particles inside. Now the first solution, the fluorescein diacetate, is added and incubated for 10 minutes. The next step, after 10 minutes, the tropedium iodide solution B is added. And mixed again. And the sample is ready to be analyzed. You see now here the screenshot of the instrument. The instrument is prepared for analysis, and then the sample is attached. Now you see that on the screen populations are formed, and by simple tools the populations are separated from each other, and we just wait until the instrument stops counting. It takes less than a minute, and after that procedure we have the result, and we can print and report the result. So this report then can be exported into an analytical report, which is then printed, or you just save that and monitor the result in a different way. Okay, yeah, I'm coming now to the end of my talk. I just would like to have a conclusion also based on a work which has been carried out by a Dutch breeding company, Elo, which tested this Pollen Viability Kit a couple of years ago. It's already a while ago. Yeah, what are the benefits? Of course, we have the results very fast. So within minutes, we have a result which are reproducible and especially objective. Yes, so we do not underlie subjective interpretation by microscopy or by staining of interpreting the artificial pollination. So that's really an objective result which can be reproduced. It's very sensitive, the test, and the samples are stable. So that is also one result out of this poster presentation here, that the samples, once prepared, can be maintained for a while. We have no incompatibility issues with the female part, which is the case for some of the other tests. And of course, it is really based on metabolic activity and membrane integrity. So here we really test two parameters which are essential for vitality of cells. And of course, we have a high statistical value, as thousands of pollen can be analyzed in a short time. Everything is connected with a low workload. It is easy and a reliable procedure, so you cannot do much wrong in this simple procedure. A relative low cost, and one part of the reaction is that what is used by the FCR reaction test, which have been since 1917 used and compared in many comparative studies with other viability and fitness testing for pollen. And by the way, the same procedure is done also for other cell types in medical research or in microbiology, where this fluorescein diacetate is used as one of the standard dyes, and especially in combination with propylium iodide to do typically viability testing on different types of cells. Human medical cells, but even also in microbiology on yeast cells or others. And of course, the test itself can be combined in one analyzer with the analysis of ploidy and genome size, which of course for plant breeders is important. Okay, yeah, this poster presentation here, let's have a closer look to the results. So here the author stresses the speed that, thousands of really pollen can be analyzed in very short time. They worked, for example, here with cyclam pollen, and they analyzed 100 pollen grains per second. And they observe a clear separation into stained vital pollen and non-vital pollen with the FDA. They showed in this diagram that they really get, depending on the batch of the cyclam pollen, really different levels, stable levels of viability. They observed also viability versus storage time of pollen. You see here that at different temperature regimes, pollen viability is affected. And their conclusions also have been overall very positive for this test method, which has been described here. In my last slide, I just would like to give a short overview about the solutions Sysmex Partec offers for plant breeding. We have, of course, the Pollen Viability Kit, which I described right now. We have really a higher number of reagents to analyze ploidy levels of plants and genome size also of plants, so where we analyze individual nuclei extracted from leaf or other plant material. And we have different type of analyzers which can handle these reagents and carry out these tests. As Sysmex Partec, we have really a long history in this field. So since the early 80s, we were the first and still are the leading supplier for solutions for plant breeding, especially for the major test of ploidy and genome size analysis, and the only flow cytometer supplier who's really offering the whole package and also the entire expertise in customer consultancy within this field of use of flow cytometry. Yeah, so I'm thanking you for your attention, and I'm looking forward to answer your questions. Thank you very much.
Okay, thank you, Matthias, for your fabulous presentations. Maybe I will start to do the Q&A sessions. I think we have received so many questions from the participants. So, okay. May you please unscreen?
Yes.
And share your screen
just
first.
A second.
Okay.
Okay.
Okay. So let me share my screen. So we will enter the Q&A sessions now. Sorry. Jadi Bapak Ibu terima kasih banyak, sudah banyak sekali pertanyaan yang masuk ya. Mungkin saya akan tanya satu persatu sesuai dengan urutan chat yang sudah masuk. So the first questions from Mr. Arif Hidayatullah from Minamas Plantations. So his question is, how do you define that pollen grain or leaf pollen grain of palm oil by using microscopy? So to determine the exact or defect samples. And is there any difference of normal pollen and abnormal pollen based on picture that contribute for abnormal seeds?
Okay, yeah, thank you for this question. Of course, you can imagine that that I'm not a specialist in palm oil pollen morphology. Yes, so for me, that question is that specific question to this type of pollen is really difficult to answer. I once a couple of years received some palm oil pollen from Indonesia, actually, and we did test our protocol on that, and I also showed some pictures during this talk from palm oil pollen. But let's say on the morphological discrimination, I have really, frankly speaking, not no experience to answer this question. Yeah.
Okay, maybe we will do the second questions then. So for the second questions, sorry, from Mr. Abdul Mutolib from London, Sumatra. So the question is it allowed to rehydrate pollen first to optimize the pollen viability? And may you share what is the procedure to do so?
Yeah, so I think the rehydration is allowed. You can do that, and the procedure usually is that you just will expose the pollen in suspension for a certain while. You put it, of course, into the, you have to put it into suspension for carrying out this test. So in our example, we used a simple PBS buffer, but you can also add, of course, some sugar components to that buffer in order to adapt it to a specific, let's say, requirement of a pollen. But here you have to look in detail, really, how this is affecting the viability of the pollen. But let's say as a first approach, I just would remain the pollen grains for a limited period of time into the suspension buffer to hydrate them.
Okay. So for the third questions, check. So the third question is from Ms. Mala from Viabilitas. Do you have any experience working with sugarcane? And what are the critical factors that affect sugarcane pollen viability? Is it the genotype or the environment factors? What are the environmental conditions that affects the most? And for how long sugarcane pollen is able to store to maintain high level of the viability? I think it's a bit long question. And what is the best of suitable preservation media to maintain sugarcane pollen viability level? I think I will just share the screen. Oh, sorry.
Yes.
Mm-hmm.
I think, of course, also this this question is very specific on the pollen of sugarcane, and of course here we also I think we have not tested sugarcane in our test. The one question, or maybe the essential question from this is, let's say, how sugarcane pollen is behaving over time. So how is the viability development? So is it reduced? When is it reduced? Under which environmental conditions? I think that is the perfect question also for this test. Yes. So here, these development of sugarcane viability could be tested extraordinarily well by using this test, as it has been done in the poster for cyclamen. But from our side, we have not investigated that on sugarcane pollen. But yeah, it's a really good approach, and if exactly those questions are addressed by these kind of tests. Yeah.
So for the next questions, you have pretty much questions here, Matthias. So the next question is from Mr. Julianus from PT Minamas Plantation. His question is how to handle pollen to maximize shelf life, and for how long can pollen be stored without affecting seed germination and abnormalities?
Yeah, here we of course have to consider a great variability of pollen of different species. Yes, so I think all these parameters which have been part in this question are need to be individually answered and investigated on specific questions. So for a specific species, I think here a literature research would help to look what is known about that, what has been already done. And for, let's say, the most popular economic agricultural species, there is some information in literature. But I think here someone needs to really go in detail for a specific species, or let's say for at least closely related species within the same family, within the same type of pollen, and draw conclusions from that information.
So for the next questions, from Mr. Kusno, PT East West in Seed Indonesia. His question is number one, how long is the storability time of horticulture pollen, for example, tomatoes, chilies, eggplants? And the second question is there any pollen release standard?
Okay, starting with the second question. So from my knowledge, there is no standard release standard for pollen, and yeah, the storability time of horticulture pollen, I think here we have to look again into the species and the conditions. So I do not really like to give here specific time frames. Definitely the viability of pollen is decreasing with time. If someone is interested in doing such investigations on a specific pollen, and we saw also from the cyclam example that this may also depend on the lot, on the conditions under which the pollen have been developed, under the environmental conditions, the genotype conditions, so that there from pollen lot to pollen lot are also significant differences. And this will definitely affect the general question for the storage of pollen. So here probably one solution would be really just to monitor that, to collect experience on the material you are working with in order then to know and forecast also, when you know what is the history of the pollen, how long this can be stored.
Okay, thank you, Matthias. We will move on to the next questions from Ms. or Mrs. Kotimato Asmi from Indonesian Vegetable Research Institute. So her question is the incubation time of the Sysmex Pollen Viability Kit the same for all sample type? So it needs 10 minutes for any plants?
Yeah, so that is, let's say, that is our proposal, to have it at 10 minutes. But so this period can be adapted also to the pollen. For example, if you have pollen which has in general a low metabolic activity, and you find out that after 10 minutes you get a weak response, a weak fluorescence signal, then there is no problem to extend that to 20 minutes, for example, in order just to accumulate more fluorochrome inside the cytoplasma and get a stronger signal. Yes. So as said, we are measuring here physiological status, and especially when you have pollen which have been strongly dehydrated before, it will need a certain time to recover metabolism, and that can be also slow in the beginning. So therefore, 10 minutes is a first recommendation, but if you see that 10 minutes are too short, it is possible to increase the time, or also if it can be possible that the samples can be analyzed already after 5 minutes. So this can also happen. So that means 10 minutes is a general recommendation, but variations are possible.
Okay. Okay, for the next questions from Mrs. Ida Febriantin from ASEAN Agri. So what are the possible aspects that might become the obstacle of a successful analysis using the Pollen Viability Kit?
Yeah, I think the major obstacle is that the pollen itself is in a status And that's the same obstacle which is valid for the fluorochromatic reaction test, so that the membrane is permeable, even though the pollen is not dead or damaged. So this can happen in certain cytological status of the pollen grain, for example, in the status of complete dihydration. So then the pollen is not active, and we will not be able to get a fluorescent signal. So in that case, of course, all pollen will be affected, and in general, this pollen then either need to be transferred from one physiological state into the other by hydration, for example, or it cannot be analyzed at all with this type. So this cannot be excluded.
Okay, for the next question. Next question is from Mr. Fateh Hisam from STPSAS. His question is, can we distinguish normal or abnormal pollen? And in terms of incompatibility, how about the accuracy?
Okay, normal and abnormal pollen, of course, we cannot distinguish if we define abnormal pollen, for example, as morphological abnormality or as a genetic abnormality. So we are not really analyzing morphology or the genotype. For example, you may have pollen with a higher ploidy. This we cannot really, this we cannot discriminate with this test. For testing a ploidy level, a ploidy analysis of pollen can be done. That's a different reagent kit and would require first a certain experimental work to define the correct procedure. But with this test, we cannot do that. In terms of incompatibility, so incompatibility, I guess, is meant the incompatibility of pollen to a specific female style or gametophyte. Of course, here also we do not test the incompatibility. I think for incompatibility testing, artificial pollination or seed set tests would work, but not the viability test.
Okay, for the next questions from Mr. Nur Cahyo from the University of Singapore Bangsa Karawang. His question is, what is the best method to preserve pollen from the field without lowering the viability index? So he is working with a plant that has a very specific time to spread its pollen. So he needs best method to extract it.
Yeah, I think here, this question is going into the direction of some other questions which we had before. So it's depending on or targeting to the individual treatment of a specific species. So I just need to, or can repeat here, so that this cannot be generalized. And here, I think that that need to be checked if there is for this specific type some experience of pollen, which is published or not, and then need to be tested specific treatments. For example, for date palm or oil palm pollen, I think that freezing, that is at least my experience, was a really good procedure to store and keep the pollen viable for a certain period of time. Yeah.
Okay. So maybe we still have at least 6 or 7 questions for you, but maybe we will postpone. So we will skip the questions to, I think I will just postpone the questions for the last session. Is it okay, Matthias?
Yes, that's okay.
Ya, oke. So, Bapak Ibu, karena waktunya mungkin diskusinya sangat menarik ya, jadi waktunya agak kurang. Jadi mungkin kita akan masuk dulu ke sesi product introductions, lalu mungkin di sesi terakhir ya kita akan lanjutkan lagi untuk pertanyaan-pertanyaan yang masih belum sempat dibacakan dan belum sempat dijawab. Jadi mungkin sedikit sesi berikutnya saya akan invite untuk Bu Amanda. Bu Amanda adalah produk spesialis dari PT Sysmex Indonesia. Jadi beliau sekarang menjabat sebagai produk spesialis di PT Sysmex. Background-nya beliau lulus dari University of Birmingham, Biological Science. Sebelumnya sempat menjabat sebagai produk eksekutif dan sekarang menjadi produk spesialis untuk instrumen flow cytometry di PT Sysmex Indonesia. Jadi mungkin dari Bu Amanda ada sedikit introduction untuk produk ya. Mungkin waktunya saya persilakan Ibu Amanda.
All right. Thank you, Stephanie, for the introduction. First of all, I would like to thank Matthias for the wonderful presentation. I think we all learned a lot today. And also thanks again for taking time from your busy schedule to support our event. Now, please allow me to conduct my presentation in Bahasa. Saya izin share screen. Baik, screen saya sudah terlihat ya. Selamat sore dan salam sehat kepada Bapak dan Ibu yang terhormat. Perkenalkan, saya Amanda Felicia sebagai produk spesialis flow cytometry dari PT Sysmex Indonesia. Pertama-tama saya mengucapkan terima kasih banyak kepada PT Elo Karsa Utama selaku host dari acara webinar pada hari ini. Dan kami juga sangat senang karena bisa mengundang perwakilan dari Sysmex Jerman untuk memberikan paparan yang sangat menarik terkait peran flow cytometry dalam kontrol kualitas benih. Sekarang, selama 15-20 menit ke depan, saya akan sharing informasi mengenai solusi yang dimiliki oleh Sysmex yang dapat membantu Bapak dan Ibu untuk proses pemulihan tanaman yang lebih efektif dan juga lebih optimal. Sebelumnya, izinkan saya untuk memperkenalkan company profile kami secara singkat. Sysmex Corporation adalah perusahaan global yang bergerak di bidang IVD atau in vitro diagnostics. Sysmex Corporation didirikan pada tahun 1968 dan berpusat di Kobe, Jepang, dengan kantor regional di Jerman, Singapura, Shanghai, dan Amerika Serikat. Sysmex Corporation saat ini tercatat dalam top 10 Global IVD Company dan merupakan satu-satunya perusahaan yang berbasis di Asia. Di Indonesia sendiri, Sysmex Corporation diwakilkan oleh PT Sysmex Indonesia yang kantornya terletak di Cyber 2 Tower, Jakarta. Sysmex memiliki coverage area yang luas karena memiliki beberapa distributor dan juga mitra bisnis. Salah satunya termasuk PT Elo Karsa Utama. Kami juga telah tersertifikasi ISO 9001-2015 dan memiliki sertifikasi CDAKB atau cara distribusi alat kesehatan yang baik. Dan untuk memastikan bahwa kami selalu memberikan yang terbaik bagi pelanggan kami rutin mengadakan survei tahunan untuk memastikan tingkat kepuasan customer kami. Dan tahun 2020 kemarin, hasil yang diperoleh adalah memuaskan. Sysmex memiliki layanan call center mulai dari pukul 8 pagi hingga 9 malam, hari Senin sampai Jumat, dan menyediakan layanan on-call service saat hari libur. Sysmex juga memiliki training center yang sudah tersertifikasi. Berikut adalah rangkaian produk Sysmex untuk kebutuhan laboratorium Bapak dan Ibu. Peralatan laboratorium yang kami tawarkan sangat beragam, mulai dari produk hematologi, koagulasi, urinalisis, kimia klinik, dan imunologi, hingga flow cytometry yang akan saya highlight di presentasi ini. Kami juga selalu berinovasi untuk memastikan produk serta layanan yang kami tawarkan selalu memberikan yang terbaik. Nah, slide ini adalah sedikit sejarah bagaimana Sysmex yang memiliki background healthcare memutuskan untuk memasuki bisnis industri. Partec GmbH adalah perusahaan asal Jerman yang berdiri pada tahun 1967. Pada tahun berikutnya, teknologi flow cytometry yang pertama kali dipatenkan, yaitu ICP-11, dibuat available untuk komersial. Selama tahun-tahun berikutnya, Partec memenangkan berbagai penghargaan untuk produk-produk flow cytometry yang dimiliki. Hingga akhirnya pada tahun 2013, Partec diakuisisi oleh Sysmex dan dinamakan menjadi Sysmex Partec GmbH. Dan di Indonesia diwakilkan oleh PT Sysmex Indonesia. Dan hingga saat ini, berikut adalah rangkaian produk flow cytometry industri yang dimiliki oleh Sysmex, yang akan saya perkenalkan di slide-slide berikutnya. Khusus untuk produk riset dan industrial, kami menunjuk PT Elo Karsa Utama sebagai mitra bisnis karena ekspertis mereka di bidang tersebut. PT Elo Karsa Utama didirikan tahun 1981 dan merupakan salah satu distributor ternama dalam bidang life science, biotech, microbiology, medical products di Indonesia. Rangkaian produk yang mereka miliki juga sangat komplit, mulai dari life science, industri, animal health, dan juga alkes. PT Elo Karsa Utama memiliki kantor pusat yang terletak di Jakarta dan 4 kantor perwakilan yang terletak di Bandung, Bogor, Jogja, dan Surabaya. Dan sesuai dengan slogan mereka, yaitu Serving You Better, mereka juga terus berupaya untuk memberikan yang terbaik bagi Bapak dan Ibu. Sekarang saya akan sharing membicarakan solusi apa saja yang dimiliki oleh Sysmex untuk membantu mengoptimalkan proses pemulihan tanaman. Untuk industri agriculture, kami memiliki solusi yang lengkap, baik dari segi alat maupun reagen. Untuk ketiga aplikasi berikut, yaitu penentuan tingkat ploidi, ukuran genom, serta viabilitas pollen. Saya akan mulai dari viabilitas pollen. Tadi sebetulnya sudah dijelaskan secara komprehensif oleh Matias, namun saya izin untuk mengingatkan kembali. Nah, khusus untuk aplikasi tersebut, kami memiliki reagen Pollen Viability Kit yang digunakan untuk menghitung mikrospora, serta membedakan antara serbuk sari yang masih hidup dan juga yang sudah mati. Reagen ini ready to use dan mengandung staining solution untuk analisa pollen dan mikrospora pada flow cytometry. Slide ini menunjukkan workflow pemeriksaan viabilitas pollen dengan flow cytometry dan reagen Pollen Viability Kit. Pertama-tama, sampel pollen yang dikumpulkan dicampur dengan buffer terlebih dahulu sebelum ditambahkan solusi A yang terdapat dalam kit. Setelah proses inkubasi selama kurang lebih 10 menit di suhu ruang, ditambahkan dengan solusi B yang juga disediakan dari kit. Dan setelah itu baru sampel dapat running di alat. Hasilnya pun keluar dalam waktu beberapa menit saja. Dan jika dibandingkan dengan metode-metode staining dengan mikroskopi yang masih sangat umum, tentu saja pemeriksaan flow cytometry akan menghemat banyak waktu dan juga tenaga kerja dari Bapak dan Ibu sekalian. Selain itu, hasil yang diperoleh juga objektif dan tentu saja lebih reliable dibandingkan dengan metode manual. Berikut adalah contoh hasil yang dikeluarkan oleh alat. Di gambar sebelah kiri, ini menunjukkan persentase mikrospora yang terdapat dalam sampel. Sementara gambar yang sebelah kanan, ini menunjukkan perbandingan antara serbuk sari yang masih hidup dan yang sudah mati. Dan kalau kita lihat contoh di gambar ini, maka persentase pollen yang sudah mati jauh lebih banyak jika dibandingkan dengan yang masih hidup. Sementara kalau yang kita inginkan tentu lebih banyak yang masih hidup ya, agar bisa bergerminasi untuk menghasilkan tanaman yang berkualitas. Untuk pemeriksaan pollen dengan reagen Viability Kit, maka Bapak dan Ibu dapat menggunakan rangkaian flow cytometry kami yang dilengkapi dengan laser biru, baik itu Cyflow maupun Cyflospace. Dalam satu kit ini terdapat 2 botol solution yang berisi solution yang dapat digunakan untuk 100 tes. Dan peran flow cytometry di sini adalah sebagai metode screening yang cepat dan juga akurat untuk mengecek viabilitas serta kualitas dari serbuk sari. Semakin banyak jumlah polen yang viable dalam suatu sampel, maka semakin besar potensi sampel tersebut untuk berpolinasi dan menghasilkan produk tanaman dengan kualitas tinggi. Sekarang saya izin lanjut ke solusi berikutnya yang dimiliki oleh Sysmex, yaitu analisa ploidi. Tapi sebelumnya saya akan memberikan sedikit background terkait ploidi. Ploidi adalah jumlah set kromosom yang dimiliki oleh suatu makhluk hidup. Dan kita sendiri sebagai manusia merupakan makhluk yang diploid karena memiliki 2 set kromosom. Dalam industri pemulihan tanaman, penentuan level ploidi sering digunakan untuk meningkatkan yield serta produktivitas. Dan juga selain itu, tingkat ploidi juga seringkali dikaitkan dengan produksi keturunan atau tumbuhan hybrid dengan tingkat fertilitas yang tinggi. Sebetulnya poin 1 dan poin 2 ini saling berkaitan karena semakin subur tanaman tersebut, otomatis tingkat produktivitasnya juga semakin tinggi. Lalu yang ketiga adalah untuk memproduksi tanaman dengan karakteristik yang ideal. Jadi kita bisa memilih ciri-ciri apa saja yang ingin kita kembangkan untuk generasi berikutnya. Berikutnya adalah untuk meningkatkan toleransi suatu tanaman terhadap faktor-faktor eksternal, seperti perubahan iklim, hama, penyakit, dan lain-lain. Yang terakhir adalah hemat dari segi waktu. Karena jika kita ingin mengembangkan varian tanaman baru, maka teknologi flow cytometry akan sangat membantu dalam memberikan hasil yang akurat dan juga cepat, jika dibandingkan dengan metode-metode breeding yang konvensional. Sysmex memiliki solusi ploidi yang komplit, mulai dari alat, reagen, hingga consumables. Khusus untuk analisa ploidi, maka kami dapat menawarkan alat Cycloploidy Analyzer yang memang sudah dirancang khusus untuk aplikasi ploidi maupun genome size. Alat ini dapat di-upgrade dengan tambahan autoloader untuk analisa sampel dalam jumlah yang lebih banyak sekali running, untuk memudahkan pengguna apabila bapak dan ibu memiliki workload yang cukup tinggi. Selain itu, kami juga memiliki rangkaian reagen yang dapat digunakan untuk aplikasi tersebut, yang akan saya perkenalkan di slide-slide berikutnya. Dan yang terakhir, Sysmex juga menawarkan filter Celtrix yang digunakan untuk menyaring partikel-partikel atau debris yang seringkali terdapat dalam sampel tumbuhan, untuk memberikan hasil akhir yang lebih tepat dan juga akurat. Filter ini diberi color code sesuai dengan ukuran masing-masing, dan khusus untuk sampel-sampel tanaman yang cukup problematik atau mengandung banyak lendir dan metabolit sekunder, maka dapat menggunakan filter dengan ukuran yang lebih kecil supaya proses filtrasinya lebih optimal. Jadi sebagai kesimpulan, Cycloploidy Analyzer adalah flow cytometer dengan konfigurasi yang sudah dioptimalkan untuk industri ploidi dan juga genome sizing. Alat beserta softwarenya sangat user-friendly dan juga mudah untuk digunakan. Cycloploidy Analyzer memberikan hasil analisa peaks yang sudah automated, sehingga hasil akhir yang diberikan sangat mudah untuk diinterpretasikan. Tadi saya juga sudah mention juga bahwa alat ini dapat digunakan dan dapat di-upgrade dengan tambahan autoloader untuk memberikan hasil throughput yang lebih tinggi. Dan jika kita lihat dari segi dimensi serta berat, maka Cyclo termasuk alat yang cukup compact dan juga memiliki desain benchtop, sehingga dapat ditempatkan di mana saja tanpa memerlukan banyak space. Instrumen ini tersedia dalam 3 versi. Versi 1 yang dilengkapi dengan UV LED, versi 2 yang dilengkapi dengan laser hijau, sementara yang versi 3 ini yang paling lengkap, yang dilengkapi oleh 2 sumber cahaya, yaitu UV LED dan laser hijau. Nah, sedikit fitur dan benefit dari Cycloploidy Analyzer. Alat ini dilengkapi dengan UV LED yang sangat optimal untuk eksitasi pewarna dapi saat analisa ploidi. Dan jika dibandingkan dengan sumber cahaya lainnya, maka UV LED memiliki lifetime yang panjang serta harga yang lebih affordable. Fitur berikutnya adalah laser hijau. Laser hijau ini sangat efektif untuk eksitasi propidium iodide yang digunakan untuk analisa ukuran genom. Laser hijau juga mencegah eksitasi dari sampel dari kontaminan klorofil yang seringkali terdapat dalam sampel tumbuhan untuk hasil yang lebih akurat. Dan seluruh alat Sysmex Partec juga dilengkapi dengan teknologi True Volumetric Absolute Counting, yang berarti user tidak memerlukan reagen tambahan atau reference bit saat menghitung sel untuk cost yang lebih rendah. Dan karena analisa peaks-nya sudah otomatis, maka akan meningkatkan efisiensi serta menghemat waktu analisa. Dan yang terakhir, alat ini memiliki desain benchtop dan tidak memerlukan tambahan PC untuk ukuran yang kompak serta portable. Selain ploidy analyzer, kami juga memiliki SciFlow Space yang seringkali digunakan dalam industri ploidy atau genome sizing. Karena Space termasuk alat yang sangat fleksibel, maka hingga saat ini ada lebih dari 20 konfigurasi yang terdaftar. Namun yang spesifik untuk ploidy atau genome adalah versi 7 dan juga versi 9 yang dilengkapi dengan UV LED dan juga laser hijau. Nah, bedanya dengan ploidy analyzer, Space memiliki autoloader yang bisa memuat 2 sampel plate berukuran 96 atau 384 well sekaligus dalam sekali running. Jadi throughput yang dihasilkan jauh lebih banyak. Dan Space ini juga memerlukan additional PC yang sudah included. Dan yang terakhir, software dari autoloader SciPath juga sudah terprogram secara otomatis untuk menganalisa histogram DNA. Dan juga laporan akhir yang diberikan adalah dalam bentuk Excel, sehingga sangat mudah dan juga praktis sekali. Ini adalah rangkaian reagen Sysmex untuk aplikasi ploidy dan juga genome sizing. Portfolio reagen saya bagi menjadi 2. Reagen yang di atas ini mengandung pewarna dapur khusus untuk analisa ploidy saja. Dan reagen ini sangat ideal untuk digunakan dengan alat yang dilengkapi dengan UV LED. Jadi ploidy yang versi 1 atau yang versi 3. Nah, sementara yang untuk bagian bawah ini adalah reagen yang mengandung pewarna PI atau propidium iodide untuk analisa ploidy maupun ukuran genom. PI ini idealnya digunakan dengan laser hijau atau bisa juga dengan laser warna biru. Dan seluruh reagen ini dapat digunakan untuk 250 tes. Yang ingin saya highlight di sini adalah reagen UV Ox Protect dan juga PI Ox Protect. Karena reagen ini sudah diformulasikan secara khusus untuk sampel-sampel tumbuhan yang mudah teroksidasi atau yang banyak mengandung metabolit sekunder. Kedua hal tersebut tentu dapat membawa dampak negatif terhadap hasil analisa ploidy dengan flow cytometry, karena sampel seringkali tidak terbaca oleh alat. Oke, jadi apa sih efek oksidasi dari sampel tumbuhan? Efek dari oksidasi terhadap tumbuhan, yang pertama, oksidasi akan merusak nukleus, sehingga akan menyebabkan sampel menjadi berwarna kecoklatan atau browning. Hal itu akan mengakibatkan efek negatif pada analisa karena memberikan hasil yang tidak akurat. Lalu produksi materi oksidan yang berlebihan juga lama-kelamaan juga akan mengakibatkan stres oksidatif dan merusak komponen-komponen sel seperti lipid, protein, metabolit, dan juga lain-lain. Lama-kelamaan hal tersebut akan berujung kepada kematian sel. Dan kalau selnya sudah mati, tentu saja tidak dapat memproduksi tanaman yang berkualitas. Oleh karena itu, reagen Ox Protect ini adalah solusi yang paling tepat untuk menghindari kendala-kendala yang tadi saya sebutkan. Nah, sebagai kesimpulan, flow cytometer Sysmex memberikan solusi yang komplit untuk analisa ploidy dalam rangka seleksi benih yang lebih optimal. Teknik flow cytometry merupakan metode yang efektif untuk memantau serta mengevaluasi kualitas dari benih. Workflow-nya sendiri cukup mudah dan juga simpel, dan waktu analisanya hanya kurang dari 2 menit. Selain itu, rangkaian reagen kami juga sudah ready to use demi kenyamanan Bapak dan Ibu. Oke, terakhir, sebagai referensi, berikut adalah negara-negara di Asia Pasifik yang sudah menggunakan solusi Sysmex dalam laboratorium mereka untuk mengoptimalkan proses pemulihan tanaman. Dari keseluruhan populasi di Asia Pasifik, negara India lah yang memiliki total instalasi terbanyak, karena sama seperti Indonesia, India juga merupakan negara dengan sektor pertanian yang sangat dominan. Selain India, kami juga memiliki beberapa referensi di Australia, Malaysia, Vietnam, Eropa, dan juga Amerika Serikat. Untuk informasi lebih lanjut mengenai portfolio flow cytometry kami, maka Bapak dan Ibu dapat menghubungi nomor WhatsApp atau alamat email yang tercantum di slide berikut. Nah, sekarang sebelum mengakhiri presentasi saya, kami juga ingin mengingatkan bahwa feedback dari Bapak dan Ibu sangatlah berharga. Oleh karena itu, kami mohon bantuannya untuk mengisi survei singkat dengan cara scan QR code yang ada di tampilan slide atau melalui link yang nantinya akan kami share via chat box. Sekali lagi saya ingatkan bahwa kami hanya akan memberikan sertifikat partisipasi kepada Bapak dan Ibu yang mengisi feedback form dengan lengkap. Baik, sekian saja paparan dari saya. Semoga dapat diterima dengan baik oleh Bapak dan Ibu. Dan sekali lagi saya mengucapkan terima kasih banyak atas perhatiannya. Dan saya kembalikan lagi ke Bu Stefani selaku moderator. Terima kasih dan salam sehat.